Hi everyone, I am Dr. C. S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and subscribe the channel. In the last session, we dealt with factors influencing enzyme activity. In today's session, we will continue further. Pharmacological action of many drugs may be explained by competitive inhibition. A few examples are given below. Sulfonamides. They are commonly employed antibacterial agents. The bacteria synthesize folic acid by combining PABA with terryl glutamic acid. Bacterial wall is impermeable to folic acid. Sulfa drugs, being structural analog of PABA, will inhibit the folic acid synthesis in bacteria and they die. The drug is non-toxic to human cells because human beings cannot synthesize folic acid. Preformed folic acid is essential for man. In this diagram, we can see that Gerard Domac was the first person to find the similarity between sulfanilamide and para-amino benzoic acid. Coming to the next drug, that is methotrexate. It is 4-amino N10-methyl folic acid. It is a structural analog of folic acid and so can competitively inhibit folate reductase enzyme. It is essential for DNA synthesis and cell division. Therefore, methotrexate is used as an anti-cancer drug. The next drug is dicumarol. It is structurally similar to vitamin K and can act as anticoagulant by competitively inhibiting the vitamin K activity. Isotonic acid hydrazide. It is a commonly used antituberculous agent. It is structurally similar to pyridoxal and prolonged use of INH may cause pyridoxal deficiency and peripheral neuropathy. Here are certain examples of the competitive inhibition. Allopurinol inhibits xanthine oxidase and is an anti-gout agent. Dicumarol is a vitamin epoxide reductase enzyme and is an anticoagulant agent. Penicillin inhibits transpeptidase enzyme and is a antibacterial agent. Sulfonamide inhibits steroid synthetase and is an antibacterial agent. Trimethoprim is a F2 reductase inhibition enzyme inhibitor and it is an antibacterial agent. Pyrimethamine inhibits the same enzyme and it is a anti-malarial agent. Methotrexate inhibits the same enzyme and is an anti-cancer agent. 6-mercaptopurine is an inhibitor of adenylosuccinate synthetase and it is an anti-cancer agent. 5-fluorouracil is an inhibitor of thymidylate synthetase and is an anti-cancer agent. Azacitidine is an inhibitor of phosphoribosyl transferase, aminotransferase, and it is an anti-cancer agent. Cytosine arabinoside is an DNA polymerase inhibitor and is an anti-cancer agent. Azocyclovir is a DNA polymerase enzyme inhibitor and it is an antiviral agent. Neostigmine is an inhibitor of ACH esterase and it is an anti-myasthenic agent. Alpha-methyl dopa is an anti-dopa decarboxylase and it is anti-hypertensive agent. Lovastatin reductase is an inhibitor of HMG CoA lowering agent and it is an anti-cholesterol agent. Oseltamivir or Tamiflu is an inhibitor of neuraminidase and it is anti-influenza agent. Coming to the next, non-competitive inhibition or irreversible inhibition. The variety of poisons such as iodoacetate or the heavy metal ions such as lead or mercury and oxidizing agents act as non-competitive inhibitors. In this diagram, we can see that enzyme when combines with substrate an ES complex is formed and which later use the product. But instead of substrate, if the non-competitive inhibitor binds with the enzyme, then a substrate cannot bind with the enzyme and hence no product is formed. There is no competition between a substrate and inhibitor. The inhibitor usually binds to the different domain on the enzyme other than the substrate binding site. Since these inhibitors have no structural resemblance to the substrate, an increase in the substrate concentration will generally not relieve this inhibition. Here is in this graph we can see that 1 upon S substrate concentration is plotted against 1 upon velocity, where Km value is the same but the Vmax value changes with inhibitor and without inhibitor. Here substrate concentration is plotted against velocity, where Km value is the same but Vmax value changes with inhibitor and without inhibitor. Cyanide inhibits cytochrome oxidase. Fluoride will move magnesium and manganese ions and so will inhibit the enzyme inorase and consequently the glycolysis. Iodoacetate would inhibit enzymes such as enzyme having HH group in their active centers. British anti-heaviside agent or dimercaprol is used as an antidote for heavy metal poisoning. The heavy metal act as enzyme poisons by reacting with the SH group. BAL has several SH groups with which the heavy metal ions can react with and therefore their poisonous effects are reduced. The inhibitor combines with the enzymes by forming a covalent bond and then the reaction becomes irreversible. The velocity or the Vmax is reduced but the Km value is not changed because the remaining enzyme molecules have the same affinity for the substrate. Increase in the substrate concentration will abolish the competitive inhibition but will not abolish the non-competitive inhibition. Coming to the difference between competitive inhibition and non-competitive inhibition. Drugs acting on active site in case of competitive inhibition but may or may not act on in non-competitive inhibition. Structure of inhibitor is substrate analog in case of competitive inhibition, but it is a unrelated molecule in case of non-competitive inhibition. Inhibition is reversible in case of competitive inhibition, but it is generally irreversible in case of non-competitive inhibition. Excess substrate, it is inhibition relieved in case of competitive inhibition but no effect in case of non-competitive inhibition. Km value is increased in case of competitive inhibition but no change is seen in case of non-competitive inhibition. Pmax value has no change in competitive inhibition but decreased in non-competitive inhibition. Significance of the in is drug action takes place in case of competitive inhibition, whereas it is toxicological in case of non-competitive inhibition. Coming to the next factor, uncompetitive inhibition. Here, inhibitor does not have any affinity for the free enzyme. Inhibitor binds to enzyme substrate complex but not to the free enzyme. In such cases, both Vm and Km are decreased. Inhibition of the placental alkaline phosphatase, that is Regan isoenzyme, by phenylalanine is an example of uncompetitive inhibition. In the diagram, in the graph, we can see that 1 upon substrate concentration is plotted against 1 upon velocity, where both Vm max and Km value are changed with inhibitor and without inhibitor. Next is suicide inhibition. It is a special type of irreversible inhibition of enzyme activity. It is known as mechanism based inactivation. The inhibitor makes use of enzyme's own reaction mechanism to inactivate it. This is known as mechanism based inactivation. In suicide inhibition, the structural analog is converted to a more effective inhibitor with the help of the enzyme to be inhibited. The substrate like compound initially binds with the enzyme and the first few steps of the pathway are catalyzed. This new product irreversibly binds to the enzyme and inhibits further reactions. For example, ornithine decarboxylase, that is ODC, catalyzes the conversion of ornithine to putrescine, which is necessary for polyamine synthesis. When the ODC in trypanosoma is inhibited, the multiplication of the parasite is arrested. Therefore, inhibitors of ODC enzyme such as trifluoromethylornithine or DFMO have been found to be effective against trypanosomiasis, that is sleeping sickness. DFMO is initially inert, but on binding with the enzyme forms irreversible covalent complex with the coenzyme pyridoxal phosphate and the amino acid residues of the enzyme. In the mammalian cells, the turnover rate of ODC is very high, and so the inhibition of DFMO is only transient. So DFMO does kill the parasite, but there is no side effects in case of patient. A similar mechanism is observed in case of allopurinol, which is oxidized by xanthine oxidase to alloxanthine, that is a strong inhibitor of xanthine oxidase. The anti-inflammatory action of aspirin is also based on the suicide inhibition. Arachidonic acid is converted to prostaglandin by the enzyme cyclooxygenase. Aspirin acetylates the serine residues in the active center of cyclooxygenase. Thus, prostaglandin synthesis is inhibited and so inflammation subsides. Coming to the next factor, allosteric regulation. Allosteric enzyme has now one catalytic site where the substrate binds and another separate allosteric site where the modified binds. Substrate and modifying binding sites may or may not be physically adjacent. Here in this diagram, we can see that enzyme has a one substrate binding site and another is allosteric binding site, which may be a activator or inhibitor. The binding of the regulatory molecule can either enhance the activity of the enzyme, that is allosteric activation, or the activity of the enzyme inhibit the activity of the enzyme, which is allosteric inhibition. In the former case, the regulatory molecule is known as positive modifier, and in the later case, it is known as negative modifier. The binding of substrate to one of the subunits of the enzyme may enhance substrate binding by other subunits. This effect is said to be positive cooperativity. If the substrate binding of substrate to one of the subunits decreases the avidity of substrate binding by other sites. The effect is called negative cooperativity. In this diagram, we can see that substrate concentration is plotted against velocity, where the Vmax value changes with positive modifier, sigmoid curve, which is normally found, and negative modifier. The inhibitor is not a substrate analog. It is partially reversible when excess substrate is added. The Km value is usually increased, but Vmax is usually decreased. The effect of allosteric modifier is maximum at or near substrate concentration equivalent to Km. But when an inhibitor binds to the allosteric site, the activity of the catalytic site is modified such that substrate cannot bind properly. Most allosteric enzymes possess quaternary structure. They are made up of subunits. Example, aspartate transcarbamylase has 6 subunits, whereas pyruvate kinase has 4 subunits. Here ends our session. Kindly like, share and subscribe the channel. Thank you.